Welcome everyone to week number 3 at Bio 120. We've got 2 good solid weeks behind us now. And this week you will actually start working on your final project. So let's see what we've got going on. First of all, last week we learned about some chemistry of life. And you may think, why do we have to learn about chemistry? This is a biology class. Because we've got to have atoms and molecules that make up cells, that make up organisms. So you need to understand the basics before you can move on to cells and organisms and ecosystems and all the rest that we will cover this term. You learn the importance of water, the fact that it is a wonderful insulator. It absorbs a tremendous amount of heat before it changes into the gaseous state, which is steam. It also has other important properties, adhesion, cohesion, the fact that it dissolves a lot of things in it. So there are lots of important properties of water. Carbon, you learned, was the most important element of life because it can form 4 bonds with other atoms and molecules and make huge, gigantic, complex macromolecules, which is what we need if we're going to have complex organisms in the world. And the organic molecules, carbohydrates, lipids, proteins, and nucleic acids, you learned about those 2 and their main functions. So the topics for this week are, number 1, classification. So classification just means that we put things into groups, right? We put them into groups so that we can predict what they're going to do in the future, so that we don't have to relearn every time we see a new anything, what it is, what it does, what we expect to happen with it, right? We just naturally do this as human beings, and lots of other animals do the same thing. And it's important because it gives us a basis for prediction for the future. And like I said, you don't have to relearn everything. So that the first time that you see a squirrel, then you understand what a squirrel is. And so the next time you see one, you say, oh, that's a squirrel. I know that it likes nuts. I know that it climbs trees. I know that it's afraid of my dog. You know, things like that. So that's why classification is important. And you go all the way from the domains, which contains 2 groups of bacteria and 1 group of other organisms. So you've got eubacteria domain, and that means true bacteria. The domain archaea, which contains archaebacteria, which actually means ancient, but we figured out that they're not quite as ancient as the other bacteria. And then eukarya, and that contains the eukaryotes, and that's everything else. So we got 2 domains of bacteria, and then everything else is in 1 domain, eukarya. Eukaryote means true nucleus. So they just give you an idea of what the cells of eukaryotes looks like. There are little compartments called organelles inside these cells. We are eukaryotic. A mushroom is eukaryotic. A tree is eukaryotic. A worm is eukaryotic. You get the idea, right? And then everything else will be bacteria. And those are also called prokaryotes. Prokaryotes, eukaryotes. Prokaryotes are bacteria. Eukaryotes are everything else. Animals, plants, fungi, and the protists, which include algae and amoeba, all those sorts of organisms. And then you're going to learn about cell structures, and this is particularly important when we get to the discussion board postings, which I'll mention in just a minute. And that means you have to know the difference between prokaryotic cells, remember that's bacterial cells, and eukaryotic cells, and that's all the other organisms. So read chapters 20, section 1, you only have to read the first section, and chapter 4, which is all about cells and cell structures, in your textbook. I would suggest you skim through these first of all and figure out where everything is before you actually start reading the intense sections in these chapters that you need. Read the Module 3 overview and watch the videos. The videos are really good about the invisible universe. That's very cool because it gives you an idea where you find microbes and domains and kingdoms of life. Also, I'm going to include this video in the announcement how antibiotics work to help you understand how they work, because you have questions about that. And then I'm going to add this other video, which I think is one of the most amazing experiments I've ever seen done. You know, I'm a microbiologist. I can't help it. Harvard antibiotic resistance in bacteria study. Just amazing. It shows you how fast antibiotic resistance happens in populations of bacteria. And let's just say it only takes 11 days for a tremendous amount of antibiotic resistance to occur. Can this happen in your body when you're taking antibiotics? It sure as heck can. Yes. So we're going to talk about antibiotic resistance. Now, this is a sea eagle that I saw in Scotland. I was standing on a boat that was pitching to and fro in the ocean water. And the sea eagle came down to hunt puffins on the rocks. You see the rocks there. And somehow, I got this photograph. Do not ask me how, because I was jumping, jiggling up and down every which way. But that's a sea eagle. And that's very rare for anybody to see a sea eagle this close up, because they're usually flying high over the cliffs. All right, so anyway, next we've got your discussion board posting, so your assignments. By the way, that's a white-fronted bee eater that I just saw in Africa in August. So your discussion board postings. Why do antibiotics target bacterial cells, prokaryotic cells, and not our cells, which are eukaryotic cells? I used to tell my microbiology students, I'd say, I can kill whatever bacteria are in you that are causing an infection. I can load you up with enough arsenic where those bacteria will die. And then, of course, they all start laughing. And they start laughing because they know if I load you up with arsenic, I'll kill the bacteria, but I'm also going to kill you. Yeah, so that's something very important about antibiotics. They have to kill the bacterial cells and not kill you and not harm you, hopefully, in any way. But sometimes they do. Yeah, sometimes they have side effects. So why do antibiotics target bacterial cells? And that's because our cells are different. And so you have to choose specific targets for these antibiotics to work and kill bacteria and not hopefully harm us. Also, you're going to answer the question, how our use of antibiotics, how the different ways that we use antibiotics and our prescribed antibiotics increases the rate of antibiotic resistance, including making superbugs like MRSA, methicillin-resistant Staphylococcus aureus, also called MRSA. But we've got other ones too. There are lots of them. So how does our use of antibiotics create more of a problem with antibiotic resistance? And then in your response post to 2 of your classmates, remember those are due by Sunday night, initial post due by Thursday night, should we be using antibiotics in livestock feed? So in the 50s and 60s, poultry farmers figured out that if they fed their healthy chickens, turkeys, etc., feed with antibiotics in them, that they grew bigger, faster, they laid more eggs, they laid larger eggs. And you're thinking, but they're not sick, so why would antibiotics affect them in any way? Because all of us fight off silent infections all of the time. Your body right now is fighting off some kind of invasion by bacteria, viruses, whatever it is. If you have silent infections caused by bacteria and you take antibiotics, the antibiotics will kill off those bacteria. And so you can use more of your energy to produce muscle tissue if you're a chicken, or larger eggs, or whatever it is. So we've been using antibiotics in livestock feed when the animals are not sick for a very long time. We're trying to get away from that practice because, of course, that leads to more antibiotic resistance. There are plenty of bacterial species and animals that also infect us. Yeah, so that's a problem. All right, and then you're also going to begin your final project. So last week you should have at least picked a topic, and I asked you to please start doing your research on it. If you didn't choose one of those topics as your topic, then you have to get it okayed by me. And if you don't do that, you run the risk this week of getting your topic turned down. And you're going to either have to redo it or you'll just get a 0 on it, because you have to do some type of topic in biology that is reasonable. Be sure to follow the Milestone 1 guidelines and rubric. All right, as a quick reminder, guidelines are instructions. Okay? Instructions, that's all that means. The rubric is how this will be graded. So you have a way to look at how this is going to be graded before it gets graded. And then if you look at the feedback on any of your assignments and my grading, lo and behold, there are all those categories again. This is also important for all of our assignments in this class. Use the correct format. So for your Milestone 1, you will put this into outline format. There's even a template in the Milestone 1 guidelines and rubric. There's a template, so use the template. There's an outline, there's an introduction, I'll show you in just a second, and then the topic. And you follow this outline. So this is not just 2 words under the bullets. You have detail, but it has to stay organized like this, and you have to label the sections. This is not an essay, okay? Not an essay. You have essays. This is not one. All right, and this is what the Milestone 1 guidelines rubric looks like. There are our topics. I already described these in week number 2. From population ecology to climate change to biodiversity, conservation, all that. Okay, so down on the bottom of this, you see here are the elements that need to be addressed. Roman numeral 1, big, introduction. All right, now look what it says. Describe the topic you selected. So you don't just list it. Genetic modification. Describe it. What is genetic modification? What is climate change? Describe it. Why is this significant or interesting to you? To the field of biology, to the world. Support your response with real-life examples. Examples are good, especially when you're presenting to an audience. They want to see why you're thinking the way that you're thinking, right? So examples are awesome. The topic section, Roman numeral 2, big Roman numeral 2. This is the second section. And then you have subheadings under this. Where? Where does this happen? If you're talking about climate change, you know, you're talking about the entire Earth. And that's the biosphere. That's every level from up in the atmosphere, down on land, to the depths of the ocean. Everything is affected by climate change. Or is it a real specific ecosystem? Is your project smaller than this? You want to look at water pollution in a river near your house, right? Near where you live. So it could be a specific river ecosystem that's affected. So where does this happen? How does it happen? How does this happen? How does climate change happen? Burning fossil fuels. You'll describe that, though, of course. Analyze it, right? So, who? What are the species that benefit from this topic? If you're talking about eutrophication, you have a lot of species that are going to be harmed by eutrophication. Everything from your marine mammals, dolphins, manatees, the seabirds, whales, whatever it is, eutrophication can harm. Fish, jellyfish, just everything. But who benefits from this? The algae that are creating algal blooms, the farmers who are allowing fertilizer to run off into the water and don't care about it, They don't care about how that's harming the environment. Industries who put extra stuff into the water that may enhance the chemistry that allows algal blooms, things like that. So who benefits from this? So those are your sections for milestone 1. Tip of the week. Put your assignments in the correct format. Later on, I think it's week 5, you're going to be asked to do that particular milestone in slide format. These are not your total presentation, just this one section you put into slides. That's the correct format. This is the outline. It tells you in the guidelines. So create an outline. Yeah, so I'm highlighting specific words in here. And then it tells you, it gives you guidelines about what this should look like. Microsoft Word document, that's fine. You can do PDF, I don't really care. Double spacing so that it's easy for me to read. I mean, that's the whole point of that. Do not make it single space, because that will drive me crazy. It also tells you the font and the size of the, I don't care about that. Just make it readable. Just make it readable for me, right? White background, black font. There you go. Reasonable margins. I will never in my life measure a margin, but just make them reasonable, right? This is not hard. And submit 1 to 2 pages. And also your references. So references is always extra, and besides the body of the work. Use in-text citations. So when you get information from somewhere, you have to include that in the text. That's what in text means, right? In the text. And then a reference section at the end, which gives the main information about that particular reference. All right, so that's my tip of the week. Put your assignments in the correct format. With that being said, I hope that you have a wonderful week number 3. And let me know if you need anything. Dr. Janie Sigmon, m.sigmon@snhu.edu. I'm here for you. All you have to do is call on me. Have a great week.